In section 9-3, we get to look at some aspects of muscle contraction. The first thing we want to do is identify some terms, and so the first two terms are tension and load. Tension is the force exerted by the muscle. Load is the force that's exerted by the object, which of course is opposing the force that's exerted by the muscle. This means that based on the degree of tension and load that is present, we can have several different types of contraction. One possible type is an isometric contraction. We can see an isometric contraction at the bottom left-hand side of our screen. In an isometric contraction, the muscle does not shorten or lengthen, but rather the tension and load are exactly balanced, and so ATP is being expended, but as those myosin heads bind to actin, they're binding to the same actin monomers each time they go through a power stroke. The tension is simply not enough to overcome the load, so the muscle remains this identical length. All the sarcomeres remain the same length, and so we refer to that as an isometric contraction. In other words, the muscle does not change in length. The other types of contraction we can have, or the other category, is an isotonic contraction. In an isotonic contraction, the muscle does change in length. And so there's two possibilities here. One is a concentric contraction. In a concentric contraction, the tension exceeds the load, and the muscle shortens. And so in this case, we see that happening in the middle of our diagram. The other possibility is an eccentric contraction. In an eccentric contraction, the load exceeds the tension, and the muscle lengthens. And so we, again, are expending ATP here, but those myosin heads are binding to actin monomers that are further and further spread out on the sarcomere. So each sarcomere is actually lengthening. We can also look at muscle contraction in terms of a single muscle fiber. So in this particular case, we're looking at figure 916 from our text on page 271. It's actually figure 916A. And we are observing a isometric twitch in this particular case. So the muscle fiber is not changing in length, but it is exerting tension because the muscle fiber cannot generate enough tension to overcome the load that's being applied by this little setup that we have here. This is very similar to the frog lab that we would have done. So here we have a muscle fiber attached to a tension measuring device. Here we have a motor neuron, and we're able to apply action potentials to this motor neuron, causing the muscle fiber to contract. A couple things we want to note in the little graph over here. One is the latent period that we talked about in that last lab. The latent period is the time that occurs, time that elapses between the time the action potential occurs and the time that we can start to measure tension in the muscle fiber. And the second is contraction time. So contraction time is the time between the end of the latent period and the peak muscle contraction. The contraction time varies, as we're going to see, in different muscle fibers. It can be as short as 10 seconds to even as long as 100 milliseconds. I meant to say milliseconds. As short as 10 milliseconds to as long as 100 milliseconds, depending on whether we have a fast or slow twitch muscle fiber. What we also notice is something that's not surprising to us, but we see that load impacts the velocity at which a muscle can contract. And in this particular figure, which is figure 918, what we notice is that we have graphed the shortening, or actually let's say the change in length of the muscle, the velocity of the change in length of the muscle on the y-axis, and then we have the load there on the x-axis. And so when the load is 0, the shortening velocity is quite rapid, and then as the load increases, we see this decrease, whereas the shortening velocity slows and slows and slows until we have an isometric contraction right here, when the load and the tension are exactly balanced. And then as the load exceeds the tension, we see that we move into an eccentric contraction. And so we see that the speed of lengthening increases and increases up till the maximum load here. So this is a result of very simply the fact that the myosin heads can move much more rapidly under no load or light load, and they move much more slowly under a heavy load. And so the movement of the myosin heads is an indicator of the speed of ATP hydrolysis. And so as the load increases, the overall speed of ATP hydrolysis is decreasing. Now, here we look at the relationship between frequency and tension. And this is what we examined, one of the things that we examined in that last lab that we had. And so if we look at this particular figure, what we notice is this, that we apply various electrical impulses to the muscle here. And when we apply the stimulus 1, we generate tension, and then that tension falls back to 0 in 200 milliseconds. And then we apply a second tension, and that falls back to 0 in 200 milliseconds. But when we apply stimulus 3, then we get the exact same tension we saw with stimulus 1 and stimulus 2. But stimulus 4 comes maybe 70 milliseconds after stimulus 3. And so we get increased tension there. And then we see, if we apply stimulus 5 and 6 even closer, we see that we get an even greater amount of tension generated than we did with stimulus 3 and 4. So in this case, it's important to realize that what we are observing is summation. And eventually, summation is going to result in tetanus, as you realize. We can see that down in the second figure. So here we have a single twitch. Here we have stimuli that are closer and closer together, creating this wavy line as the tension goes up and down, up and down. We refer to that as unfused tetanus. And then eventually, what we see is that we achieve fused tetanus if we deliver those stimuli close enough to one another. You were able to observe this with your group in that last lab. Now, the reason for this is completely different than the reason that we observed for the summation that occurred in a postsynaptic cell. It's the same term, but it's a different occurrence entirely. So the summation that occurred in the postsynaptic cell was the result of the cumulative charge that was generated as a result of excitatory and inhibitory stimuli that were applied to the cell. In this particular case, the summation that's observed in a muscle cell is entirely the result of the accumulation of calcium. This mechanism can be explained by thinking back to what we talked about in 9-2. So every time an action potential arrives, enough calcium is released very briefly that such that it binds to troponin and moves tropomyosin, exposing all the myosin binding sites on actin. So all the myosin binding sites are exposed every time that an action potential arrives. However, they're only exposed very briefly. The calcium levels start to go back down very rapidly, and that means that some of those troponin and therefore tropomyosin molecules move back and cover up those myosin binding sites on actin very, very quickly. The result is that all the myosin does not have a chance to bind to actin every single time an action potential arrives. But summation occurs when, as second action potential arrives, before all the calcium has been pumped back into the sarcoplasmic reticulum. You can see that between stimuli 3 and 4 there in the first part of our figure. And so eventually, of course, you achieve tetanus when the calcium levels in the cytoplasm of the muscle cell are high enough that all the troponin is bound, moving all the tropomyosin out of the way, so all of the actin is exposed and myosin can bind to it, and you're not going to get any more tension than that. So you can only achieve so much tension, because when all the myosin heads are bound to actin, that's as much tension as you're going to get. So what we see is that the tension that's generated in tetanus is 3 to 5-fold greater than the tension that's generated in a single twitch. And all of that is the result of the increasing levels of calcium that we observe in the cytoplasm of that muscle cell. So the last topic we want to cover here in section 9-3 is the length-tension relationship we observe in muscle fibers. Now there's two things that are depicted in this figure from our text. One is passive tension, which is shown in red. And so this is in a relaxed muscle fiber that's obviously not contracting. And what we notice is that we do generate some tension as we stretch the muscle and further and further and further. So this L of 0, as we're going to mention in just a moment, is the ideal starting length for contraction of a muscle fiber. And so in practical terms, it's typically the length that a muscle fiber possesses when it's in its relaxed state in vivo, in other words, within the organism. And so as we begin to stretch a muscle, then what we notice is we generate more and more tension as a result of various elastic, somewhat elastic at least, protein fibers that are found within the muscle fiber. But there's nothing too spectacular there. It's just the muscle fibers have a bit of elasticity, so as we stretch them, we generate tension. Mostly what we're going to focus on in this diagram is the active tension, which is shown in the green line. And so on the y-axis, we have the tension that's generated, and on the x-axis, we have the percentage of muscle length. What we'll notice is that at the L of 0 that we described a moment ago, we get maximal muscle contraction. The muscle tension is described as percent of the maximum tension you can generate, so we're at 100% at L of 0. As we stretch the muscle, the degree of tension we can generate goes down and down and down and down until when we get to 175% of the L of 0, we get no contraction, no tension generated at all. We can explain this by looking at these little drawings in the diagram. As we stretch the muscle, there's less and less overlap between the myosin heads of the thick filaments that I'm pointing to here and the actin filaments. And so eventually when you get to, although it's not shown here, you can imagine when you get to 175%, there's simply no overlap at all between the myosin heads and these thin filaments, and so no tension can be generated. On the other hand, if we go the other way and we shorten the muscle, we also see that the amount of tension the muscle can generate goes down and down and down and down until we get to about 60%. There's no tension at all generated. And so the reason for this is twofold. One is simply that as these thin filaments overlap, the myosin heads have a difficult time gripping both the actin monomers on this inside thin filament and outside thin filament. And so that cuts down dramatically on the amount of tension that can be generated. And then eventually we run into the following problem. The thick filaments actually bump into the Z discs, as is being shown here. So eventually at 60% length of L of 0, you get no force generated whatsoever because the sarcomere simply can't contract any further. The thick filaments are butted up against those Z discs. And so this illustrates this so-called length-tension relationship in skeletal muscle cells. Every skeletal muscle has an optimal starting point, and that starting point is roughly the position in which that skeletal muscle is held in a relaxed muscle.